Thank you, Mari. Thanks a lot. Hello, everybody. Mari said it earlier than I did. I wish I hardly use such words like immortals and zombies in talks, but so I wanted to take this opportunity that talk a little bit zombies as well. Well, who hasn't thought about living forever, right? Especially if you consider the other opportunities there. But let me see if I can work with this. But let's go to our cells. That's maybe more scientific for today. And if you look at the simple cell cycle, right? Our cells are practically capable of living forever. No, I mean they replicate their DNA in the. Oh, yeah, okay, I won't do this, it's too complicated for me. During S phase, and then they divide in mitosis, right? And this is kind of two identical cells, and the life goes on, and they live happily forever. But our cells don't always divide, so they have some sort of control, and they need some sort of growth signals to tell them that, yes, proliferate, go on, do something. There are important built-in checkpoints that I will talk a bit later that tells the cell that everything is actually ready for it to progress in the cell cycle. And I don't know whether you learned about this already, but there are also some limits on repeated cell divisions. That is, it's the end of the telomere is the end of the chromosomes that is getting shorter and shorter by every replication, unless there is an enzyme that takes care of it, that this doesn't happen. And cells can't always just divide, can't keep on dividing. And there are a couple of outcomes that if they don't divide, one of them is that they can grow old, like we are hopefully getting old as well at some point, and they go into some sort of cellular senescence. It's a kind of a vegetative state that they don't anymore proliferate. They stay there, and normally these cells may be taken care of the immune system that eradicates them to kill them. And I'm sure you have heard about programmed cell death. That is a program of the cell itself that it says, okay, this is enough, and then they kill themselves practically. And if this, so this is something now about this checkpoint that I said. And just a few things that, like, there are these cyclin CDK complexes at every border of the cell cycle, and they make sure that everything is right for it to happen, and if it's right, then it happens. And what's the most problematic for every cell division? Well, it's usually the DNA lesions, the problems with the DNA, and this is a huge number that in every single cell in our body, there are approximately 10 to 100,000 lesions a day that each cell has to take care of and make sure that this is kept, the DNA is kept intact. They somehow have to repair it. And if there is a problem with the DNA, then they signal these cyclins and CDKs that do not go on, stop this cell cycle, and to take care of this problem. But sometimes there's just too much of this damage that they cannot take care of it. And in those cases, they have to make such decisions what to do, and then induce cell death. So the cell would die. And there's another kind of possibility that the cell goes into some sort of cellular senescence, which I mentioned before, which they don't die, but they persist there, but they never anymore normally divide. And if this goes wrong, then we have an immortal cell practically, and the cancer cell is born. And I won't read it out, but you may read this. I like this quote from this book, The Emperor of All Melodies, that while we all want to be maybe somehow immortal, this is what the cancer cell actually achieves quite well. And I told you these huge numbers. These are not mutations themselves. These are just problems with the DNA that the cell has to take care of, and mostly it does. Nevertheless, during the lifetime of a person, 1 in 2 people develop cancer. So this is a huge, huge, huge number. I mean, not everybody has to die of cancer, of course, but so this is a real problem. And what to do with it, right? I mean, there are a couple of approaches I think we are all aware of. There's surgery, right? You just cut it out if you can. I mean, what you see, you can cut out. you get rid of. But there are often microscopic cells that already spread somewhere that you can get rid of with just surgery, and they can grow again, right? They are immortal. And there's chemotherapy that kills typically fast-dividing cells, but they also have a drawback, right? Because we have other cells in our body that divide, and it doesn't do good. And I'm sure most of you have seen people after chemotherapy. So this is pretty hard on the body, and not something really well tolerated. And then I think there's this holy grail of medicine, generally speaking, personalized medicine, right? If we know what's the problem, we can just treat the problem. That if we know this cancer carries a particular mutation, then we can maybe target this particular mutation. But I think it really needs two things, and that's for research, I think, still ahead for most of the cases, that we need to have an exact problem, and we have a therapy for it. We have to have a drug for it. That's not always the case, unfortunately. And this is now kind of going more into the territory that we work on, is that there is a mutation that actually two genes, BRCA1 and BRCA2, and mostly affect women. And just look at the prevalence of ovarian and breast cancer in the normal population, 2 to 12%. But if any person carries this mutation, the probability that they will develop cancer is huge. It's a huge, huge, huge problem. And maybe luckily for some of the people, in 2005, there was a discovery there are so-called PARP inhibitors. They are inhibitors of an enzyme called PARP1. PARP1 is an enzyme that recognizes problems in the DNA breaks. And if we add this enzyme, this inhibitor, then it gets trapped on this DNA, on this broken DNA, and only those cells can repair it properly, can deal with this problem, if they carry normal BRCA1, BRCA2 genes or gene products. And if the cell is mutant for BRCA1 or BRCA2, then the cell can't repair it, they will die. So this is great. I mean, so you have a healthy body, most of the cells are fine, you treat them with the inhibitor, nothing happens, but the cancer cell, they just go to hell. So that's what we want. Perfect. And this is what we want to understand in the lab. And the project is spearheaded by Roberta, who's also a junior mentor in the program, and she's somewhere around. I sent her back because the view is better from the back, maybe. And one of the genes, two genes that we have been working on is polymerase epsilon 3 and 4. You don't really have to know much about it, but I still tell you something about it. That you heard about replication, I'm sure, and these protein products of these genes participate in this replication. They are part of the DNA polymerase epsilon, which is kind of that light blue enzyme on the left upper part. And this is important to take care of like proper replication. That replication, the new DNA synthesis goes properly. And if these genes are mutated, and what you can maybe appreciate that the blue and the red lines are how many cells survive at a given concentration of a PARP inhibitor. Olaparib is a PARP inhibitor. And you can maybe appreciate, just take like the number 10 micromolar olaparib. I mean, the white type, healthy cell, most of it is still alive, happy, but most of these cells are dead, right? I mean, this is like, we are down to almost like very close to zero, but I think that's the problem that we are very close to zero, but this is not zero, right? I mean, very close to zero is still something other than zero. And if we take a look at these cells, the upper part are white type cells. These are the healthy cells that without treatment or after treatment of this PARP inhibitor, they are kind of like happily living in the petri dish. They are dividing, and if you look at the cells at the bottom row, that's the mutant cells, that they look quite happy without the treatment, but after PARP inhibition, I mean, there are much fewer cells, maybe you can appreciate, and if you would take a closer look at these cells, you will find that many of these cells are enlarged, flattened, and sometimes they have multiple nuclei, and just take my word for it because I won't show it now here. They are positive for beta-galactosidase, and they start to express some inflammatory signals too, because normally like they attract the immune system to probably get rid of them. Okay, so this is great. I mean, like senescent cells don't divide, so we killed most of them and the others are just vegetative. So, but
one more.
We went on.
Sorry, one more minute and then the questions.
I have one. Yeah, I'm close to it. So we have, like, if we release the cells from this inhibition, then what we find that actually these cells start to divide again. So these are practically like zombies, so they didn't die completely, and after kind of the drug is not anymore there, they actually start to live on and proliferate again. And so we tried these senolytics, these are drugs to kill senescent cells, and actually we are relatively okay with that the mutant cells, now most of them upon the senolytic treatment, half of them are dead. So maybe there is a promise that actually these cells, in compared to wild type, where senolytic doesn't do much, we can maybe actually get rid of these cells as well. But just take the quote from Jurassic Park, rest assured that life will find some way and new mutations will arise. And I think there's plenty of work to do, even there will be plenty of work for you as well to do, that we have to understand how to kind of get rid of these cancer cells, find their weaknesses, and we have to find new drugs to treat them. And let me thank the people in my lab. We also have a student from the National Academy. No, not, how do we call these? Scientist education, Zsófi, and of course our work wouldn't be possible without funding from the Hungarian Research Network, also the governmental funding, and from the academy as well. So thank you for your attention.
Thank you very much for the talk, and now we have a couple of minutes for questions. So you could see, so the lab's main focus, how to fight zombie cells. Any questions, any comments? Yes.
Thank you for the presentation. My question would be, like, in the last few minutes you mentioned that new mutations will arise in time. Do you think creating drugs that prevent or can treat mutations which we know of currently, do they help the immune system create these new mutations, or are they absolutely apart from each other?
So you have, I didn't quite, so you want to make a drug that the cell has less mutations? That's what you said? I didn't.
No. My question would be, so cancer cells, well, there are quite a few types of them, and they come from many mutations. But,
yeah.
So if someone's treated with many drugs, I don't know, has had plural cancer cells in their body before and has been treated by multiple drugs, does that help new mutations
to arise?
Yeah.
But yes, I think that's the problem. I think with chemotherapy as well, that most of them actually induce damage, and that's how they practically kill the cell, that there is just too much damage that the cell cannot deal with, and then since mostly proliferating cells are sensitive to that, and cancer proliferates. But of course, yes, and that's the problem, that many times chemotherapy ultimately over kind of many years, not every mutation leads to cancer, obviously, but some of it could, so it's bad luck, right? And then eventually this kind of can happen again, yeah.
Thank you.
Other questions?
Maybe if I may ask a question. You were referring just in your last slide, potential combination therapies. Have you tried PARP inhibitors in your system to combine with other well-known agents and to see if the potential of PARP inhibitors can be enhanced by that strategy?
Well, I mean, in some cases we might have tried. I mean, I would say that we are also mostly interested in basic research, really not really trying to help anybody in the short term, I think. But it will be very technical. I mean, like ATR inhibitors, and there are kind of like things that these are very important enzymes to signal the cell during replication that there is a problem. ATR is one of these kinases that raises a response, and if we inhibit that, cells are typically very sensitive. Roberta tried, I mean, this is very new, so Roberta tried these senolytics, look like quite promising, so if we give it in combination as well, now with PARP inhibitors, looks like many more cells die. So, yeah, but generally, yeah, so we kind of know that we haven't really done so many kind of combination therapies, I think, more broadly.
Thank you very much for the talk, and now we are taking a photo. A photo.
Please don't go away.